Hi, in this video I'm going to explain how to understand a pedigree, and below is a pedigree for a family with an autosomal recessive disease. And today we have 3 questions to answer. So as usual, I recommend you to stop video here, read the questions, try to answer each question, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. So here's the first question. What is the genotype of individual A at the disease X locus? Use plus to indicate the wild type allele and minus to indicate the mutant allele. So we can get 3 different genotypes. One can be homozygous dominant, another one can be homozygous, another one can be heterozygous, so plus and minus, and the last one can be homozygous recessive, so both mutant alleles, and we use sign minus for each allele to indicate this variant. So these two would be phenotypically normal, and this variant means that genetic disorder would be expressed. And if you take a look at this pedigree, you can tell right away that these two individuals, here this square stands for the male and this circle stands for the female, have this genetic disorder. So both of them belong to this genotype. So phenotypically would express this genetic disorder. So their genotype would be minus, minus, and here minus, minus. So because this person has both alleles, that is minus, minus, and that means that one allele he should get from his father's side and one minus allele he should get from the mother side. So each of the parents should have this allele. And because both of them are phenotypically normal, that means that the genotype would be plus and minus. So they both would belong to this genotype here and would be heterozygous and would be carriers. And now we can answer. So this would be the answer for the first question. Genotype of the parent A would be plus and minus, or heterozygous. And second question, what is the probability that an individual B is a carrier of the disease X? And the probability would be as follows. Because we know the genotypes of the both parents, it would be easy to predict the genotype and probability of the individual B to be a carrier. So a mother's genotype would be plus and minus. So we just have to build a Punnett square. And father's genotype also would be heterozygous, plus and minus, and here is a sign for the male. And as usual, when we build a Punnett square, we can predict the outcome of such a cross. And as you see here, we have plus plus, or homozygous dominant genotype. Here and here we have heterozygous genotype, and here we have 2 mutant alleles, and that means that chances 1 out of 4 that such a cross would produce genotype where this genetic disorder would be expressed. But as you see here, we have normal female, and that means that this female has 2 chances out of 3 to have this to be a carrier. Why not 2 out of 4? Because as you see, this female can belong to these genotypes. So she has 1 out of 3 chances to be homozygous dominant and 2 out of 3 to be carrier or to be heterozygous, belong to the heterozygous genotype. Why not out of 4? Because this genotype we can see phenotypically, but this we would see normal phenotypically, but cannot tell the difference between homozygous dominant and heterozygous. So we know that the genotype would be one whether homozygous dominant or heterozygous, and the question is, what is the probability that individual B is a carrier of the disease X? And the answer would be 2 chances out of 3. So 2 out of 3. And this is going to be an answer for this question. And question 3, individuals C and D decide to have a child. What is the probability that the child will have a disease X? So here we have individual C, male, and here we have individual D, female. So in order to predict the probability for the child to have this genetic disorder, first we have to find the probabilities for them to be carriers. These 2 persons cannot be affected with this genetic disorder or cannot belong to this group, because otherwise the signs would be filled with a color, and because the signs doesn't filled with a color, we know that they belong to these 2 groups. But what is the probability to belong to these groups? So for this person, we know that his father is minus minus, so he must have 1 allele that is also would be minus, and because his mother is outsider, not a part of this family that has a genetic disorder, so her phenotype should be wild type, so both of her alleles should be normal, because this is rare genetic disorder. So we can say that a genotype of this person would be plus and minus or heterozygous, and the same would be the same rule would apply to his sister, so his sister also should be carrier. And as for the this family here, we see that mother affected with this genetic disorder, but father should be phenotypically normal and genotypically normal, so should have 2 normal alleles, and that means that all the children here in this family would have 1 normal allele from the father, and mother, because has only 2 recessive alleles, that means she would provide this recessive allele for each child. So all the children in this family would be heterozygous and would be carriers. And what is the chances? Both parents here heterozygous is 100%. We can predict, looking here at the parents, that this individual would be heterozygous, and the same here this individual D also should be heterozygous. And we can use the same table here to predict the probability for their child to be affected with this genetic disorder. And as you see, according to this table, when we have two heterozygous parents, the probability would be 1 out of 4. And this is going to be the answer for the last question. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.